Hi, good afternoon, good morning, good evening, everybody. And first of all, Happy New Year 2023. All best wishes for a healthy and successful new year. I hope this new year will bring a lot of joy, happiness, but also peace to all of us. As usual, I have a special thought to all patients and families struggling with difficult and hard to treat malignant diseases. So hopefully 2023 will be a symbol of cure. I'm Mohamed Moti from the Sorbonne University and Saint Antoine Hospital in Paris, in France. And on behalf of all the International Academy for Clinical Hematology steering committee and the teams and members, it is my great pleasure to welcome you to this first webinar of the year 2023. In 2023, we will be celebrating the 5th year of the ICH activities, which were actually launched in 2018. And I would like to say a big thank you to all of you who have been supporting strongly the ICH activities. And I can assure you, in 2023, we will also have new and great initiatives, and you will continue to enjoy education in hematology without borders, because that is the founding principle and the DNA of the ICH. And for the first webinar of the year 2023, we are on the 3rd of January 2023. It is now a little bit after 6 p.m. in Paris, 5 p.m. in London, 9 p.m. in Seattle, noon in New York, 7 p.m. in Jerusalem. Our first lecturer for this year is Dr. Folashade Otegbeye from the Fred Hutch in Seattle. She is a clinical researcher and physician specialized in cellular therapy and blood stem cell transplantation. She's an associate professor at the University of Washington. And actually, in her role as the facility director of the Hutch Therapeutic Product Shared Resources, she works with the Hutch Cellular and Immunotherapy labs to translate the research ideas into a clinical product. And I think I don't need to spend a lot of time to convince this audience that the advent of cellular immune therapies has been a huge and great breakthrough in the field of hematology, but probably it's going to be beyond hematology. And actually, the logistics of the cellular therapies, especially CAR T cells, has been always a difficult topic. And the topic of Dr. Otegbeye today is about, the title is Bench at Bedside Models to Facilitate CAR T Cell Supply Chain. I think it's a really very important and very timely topic to start the year with, because these CAR T cells are bringing a lot of hope to many patients, but access, availability, and affordability is always a matter of concern. And we at the IACH, we always advocate for affordability, accessibility, and availability. So without any further ado, I'd like to welcome Dr. Otegbeye to deliver her webinar. You will have the opportunity to ask questions. So please use the Q&A box or the chat box, and we will reconvene, let's say, in around 30-35 minutes, depending on the length of her talk. So, Dr. Otegbeye, the floor is yours. See you later.
Hello, everyone. I am Dr. Folashade Otegbeye, an associate professor with the Bone Marrow Transplant and Immunotherapy Program at Fred Hutchinson Cancer Center here in Seattle, Washington, in the United States of America. I'm also the facility director of the Therapeutic Products Program here at Fred Hutch Cancer Center. I am deeply grateful and honored by the invitation from Professor Moti to have this discussion with the IACH community on a topic titled "Bench at Bedside Models Facilitates CAR T-Cell Supply Chain." The reason for this discussion is based off of a commentary I published in the TCT journal this past April on a subject: bench at bedside models facilitate CAR T-cell supply chain. And essentially, this was the bottom line article in the review of another full-length article that was published at the same time in the journal. The subject of that article was point of care anti-CD19 CAR T cells for treatment of relapsed and refractory aggressive B-cell lymphoma that was based on the experience of a group out of Israel. Manufacturing CD19 CAR T cells using lentiviral vectors produced from an NCI cell line. The idea behind my viewpoint on this article was to shed the light on increasing data coming out of different point of care models of CAR T cell manufacturing with delivery of care at the sites of at the clinical sites where these patients are being seen, and to advocate for a model, or to create some discussion advocating for models where CAR T cells are manufactured at the clinical sites where the patients are being seen, to increase the resource availability of access to this treatment platform. Now, to start, I will say that as for the disclosures, I have no employment, financial, consulting, or other relationships with any commercial entities that I will describe in this talk. In addition, I have no conflicts of interest in general, and all of the views that are proposed in this discussion are my own and do not represent any organization that I belong to or professional organization that I belong to. Now, one of the points that I raised in this article was that broad access to commercially available chimeric antigen receptor T cell or CAR T cell products for non-Hodgkin's lymphoma, acute lymphocytic leukemia, and multiple myeloma is constrained by the limited capacity of centralized manufacturing models, which is the current paradigm with the commercial CAR T products. And I mean, I know that the audience that is listening to this talk is familiar with what the CAR T cell manufacturing looks like. So this talk isn't designed to go into detail with what CAR T cell manufacturing looks like. Neither is it to go into details with the specific CAR T cell products and the efficacies or of those products specifically, because again, this is an audience that is very familiar with this product. However, to give a general overview, as we know, the production, the manufacturing process for CAR T cells starts from the collection of the starter material, which is the apheresis product from the patients. In this setting too, I'm talking about autologous CAR T cell products. So first with the leukapheresis, and then the selection of the target population, which is the population that is the T cell population that is the focus of the transduction, as well as the treatment material, the end product. So selection of those cell subsets, either by magnetic cell separation or of other processing that is done at different sites, and then activation of those cells of the CD3 cells, followed by transduction with viral vectors. There are some clinical trials ongoing looking at non-viral gene transfer methods as well. And then the expansion of those CAR T cells using different platforms, with the final product being harvested and formulated for cryopreservation, typically for the commercial CAR T products. But there are fresh infusion, there are products that are infused fresh, particularly in the point of care models, which are all currently done under IND. The cryopreserved products are then transported from the site of manufacturing, which for the commercial products are centralized facilities or centralized factories. And then those products are shipped to the treatment sites where they were previously collected from the donors, which are the intended patients, and then administered at those sites. The currently available commercial CAR T cells are manufactured currently by 4 companies, and for overall between the products that are intended, the CD19 products intended for non-Hodgkin's lymphoma and acute lymphoid leukemia, and the myeloma and the CAR T cell products targeted for myeloma, there are 4 companies that have manufacturing sites all over the world, but the approved treatment centers for all of these companies span over 300 sites all over the world. And so, to give you some perspective of the current manufacturing scope that is available for the Novartis product, which is the Kymriah or tisagenlecleucel, there are about 5 to 7 sites for manufacturing across the globe, with 5 of them being the active ones for commercial manufacturing at this time. For the Kite of Gilead products, there are 3 sites, with 1 of them being new. And again, this is a company that manufactures both the Escada as well as the Tecartus products. For the Kite products, 1 of those sites is in the US, with the other one being with a new one in development being in Maryland, also in the US, and then another site in Europe, which is in Amsterdam. For Bristol Myers Squibb, with the production of Breyanzi and currently Abecma, it appears that there are 2 sites, 1 for the Breyanzi product and 1 for the Abecma product, with a proposal to add manufacturing sites to these facilities. For Carvykti, manufactured by Janssen, now Legend Biotech, in collaboration with Johnson & Johnson, there are 2 manufacturing sites, with 1 of them being with 1 active and the other under construction. Now, so what does this mean? Of course, this means that for patients to receive this commercial CAR T products across the world, they have to be identified and then enrolled into the programs, and then the different sites, again, over 300 treatment sites across the world. These different sites have quotas for being able to get their patients enrolled into the program, and then go into a queue that is assigned for manufacturing before those products are shipped. What that means is that there is a variable turnaround time from patient identification to patient collection, and then the product manufacturing before the eventual delivery. So one of the things that has become increasingly clear from the site of care models, or the point of care models that are increasingly being published and discussed at various meetings, is the fact that the shorter apheresis to infusion turnaround time in the site of care products is also associated with reduced need for bridging therapies, as compared to where you have centralized manufacturing for commercial CAR T products. In the manuscript that I wrote this commentary on, which is published by Kedmi et al., again from experience in Israel, the median turnaround time from apheresis to infusion was 10 days. This meant that 92% of patients on that study received their CAR T infusion without needing any bridging therapy. To compare this to the Transcend NHL 001 study, in which the median time from leukapheresis to the availability of lysocell for shipment was 24 days, with a range of 17 to 51 days. And overall, a median of 37 days from apheresis to actual infusion, with the range being anywhere from 27 to 224 days. Of course, this meant that these patients would require bridging therapy, oftentimes more than one line of bridging therapy before they could receive their CAR T product. And this comes with its associated risks from the treatments that are administered. The US Lymphoma Consortium study of Axicell used as a standard of care also showed that while the time from leukapheresis to start of conditioning therapy was somewhat reduced, but however, the median time was still roughly 21 days, with a range from 11 to 71 days. As a result of this, as well as some other factors, 7% of 298 patients in this study, that looking at the patients receiving for standard of care, meaning as when it was commercially available, 7% of these patients did not receive their intended product due to either disease progression or death in the interim while awaiting the product. A similar study looking at the standard of care Axicell and Tisocell in French centers showed that about 87% of those patients received a median of 2 cycles of bridging therapy prior to receiving the infusion of the CAR T product. The other thing to note, looking at or comparing the point of care models to the commercial products, is that manufacturing efficiency does not appear to be compromised in the point of care models. With looking at a variety of studies of commercial standard of care products showing a 3 to 13% manufacturing failure, while in the published studies and then also in discussions at meetings, there's anywhere from 0 to 4% manufacturing failure in the site of care models, which again just adds to the fact that it is possible in the setting of, it's very possible in setting to maintain good manufacturing efficiency in the site of care or point of care models. In addition, the other highlight in this in my commentary on this paper was that there is comparable treatment efficacy between point of care models as well as the centralized manufacturing with commercial CAR T manufacturing. In the study of reference, the overall response rate was 63% at 4 weeks after the CAR T cell infusion, with 39% complete responses and 24% partial responses, and a 1-year overall survival of 67% for patients who had a complete or partial response. For this product, which is a CD28 costimulatory signal, in comparison to Axicell as standard of care, the overall response rate was 82% and 64% complete response rate. While I mean, this was not a head-to-head comparison between the Kedmi et al. Study as well as the standard of care Axicell. However, the patients in the Kedmi et al. Study appeared to be more significantly advanced or be more treatment refractory than the patients that were reviewed in the standard of care study. Another study that I highlighted in the commentary was a publication by Martian et al. Looking at multi-site models of again point of care, but using the same CAR T cell constructs with the 4-1BB ligand in a site in Russia and a site in the US, which was in Cleveland, Ohio, where the adult non-Hodgkin's lymphoma patients in that study had an 83% overall response rate with 70% complete responses and 93% 1-year overall survival. This was also comparable to what was seen in from lysocell and Tisocell in the Transcend NHL 001 and Juliet studies respectively, which again use a 4-1BB ligand co-stimulatory domain for the CAR T construct. In addition to this, the toxicity profiles between products manufactured at point of care settings have been very comparable to the products to commercial CAR T products, including in the trial experience for those products. So the treatment efficacy does not appear to become treatment efficacy, manufacturing efficiency, and toxicity profile does not appear to be compromised in the point of care models compared to the commercial CAR T, commercial and centralized CAR T manufacturing. Now, another highlight, or another thing that I highlighted in this commentary was that one of the constraints to site of care manufacturing models include the need for cGMP production environments and personnel, including for production and quality control, to meet the appropriate regulatory requirements and ensure product quality across sites. And this is particularly important when we start to think about different point of care centers doing their own manufacturing, and then trying to understand what is the efficacy and what is the safety profile and what's the manufacturing efficiency across these different sites when we try to do clinical studies in the future. And so to facilitate this, again, I will not go over this cartoon for manufacturing, but when you think about all the different steps that are involved in manufacturing and how to increase the possibility of doing this in a site of care model, then we have to think about manufacturing streams that move from open processes, which is the traditional manufacturing stream that is part of the development of all of these CAR T products. So open processes with mostly manual operations from the start of manufacturing all the way to harvest, to more closed or semi-automated manufacturing streams that employ several modular equipment in a closed enclosed settings, but still requires a lot of it still requires a lot of manual handling, and also which means that also requires a lot of experienced production personnel to ensure that the products remain within that the manufacturing maintains good quality. And then moving from that to the models that facilitate the bench at bedside models, which is having closed, fully automated systems, with the two prevailing examples at this point in time being the Clinimax Prodigy platform by Miltenyi Biotech, which is on the left panel, on the right panel here, and the top right panel here, and the Cocoon platform, which is developed by Lonza, which are both fully automated systems where starting from the leukapheresis product, you have fully integrated systems for cell selection, cell activation, the transduction, expansion, and the final fill of the harvest product, either for cryopreservation or for infusion. And so to ensure that the ultimate goal with the bedside manufacturing models is that you want to be able to facilitate, you want to be able to ensure the rapid delivery of these products to the patients who need them in decentralized models. But you also need to reduce the labor intensiveness of this approach and minimize intersite variability, like I mentioned earlier. And so to do this, some of the things that will facilitate this is to start with automating the key central operations, ensuring closed system manufacturing, standardizing the reagents that are used in manufacturing or the materials used in manufacturing, as well as standardizing the product identity assays. In general, also product quality control assays, but in particular, product identity assessment. Now, to give an example, again, using an example with the Clinimax Prodigy, the T-cell transduction process, where on the Clinimax Prodigy, you can start for a 10 to 14 day manufacturing, starting from preparation of the starting material, cell separation, the activation, transduction, and expansion, and then harvesting the final cell product. With the program on this platform enables reduces the hands-on time with the material and on the instrument to roughly about 4 hours over a 10 to 14 day period of manufacturing. So with this model generating sufficient CAR T cells for products to meet the doses specified in several clinical trials. There is a new platform, the TCT LC model, which is the expanded, which is designed to meet the increased cell dose that is required for engineered TCR T cell manufacturing. The idea behind this is that the more we have this kinds of platforms where we can minimize the hands-on time that is required, ensure that there is more standardized process for the manufacturing itself, it reduces the labor intensiveness of this approach and also allows side-by-side manufacturing or processing to go on in the facilities where this Clinimax Prodigy instruments are held. And so what are the things that are needed to ensure the sustainability of the bedside manufacturing models? Again, to summarize, you need to standardize and streamline. And the standardization starts from even the materials that are used. So having models where one of the things that I propose or that I think about in this setting is that the commercial product in the bedside manufacturing model would not be the CAR T, would not be the final CAR T, rather it will be the gene transfer product, which either it's a lentiviral construct or the viral DNA genetic material that is used for electroporation, for instance. So the materials, we need to standardize the materials and have them be uniform, or at least be available to be uniform across different centers. Also, the apheresis, the quality of the apheresis product, the cell dose that the starting cellular material, we need to have some form of standardization across centers. The reagents and the buffers used for cell selection, for the media, for the product manufacturing, the cytokines also, we need to make sure that we can standardize them in a way that does not require high level quality control for establishing lot-to-lot stability and ensuring that there's not significant lot-to-lot variability that would require new validation of the manufacturing system. Again, the materials that we can also standardize include the activation beads, where there are commercially available activation beads like the Dyna beads manufactured by Gibco, Transact on the Miltenyi biotech platform, as well as the Clouds beads by Biotechne. We also need to standardize and streamline the process, which I went over in the previous slides, where we need to ensure that we have closed systems for manufacturing with platforms or with programs that are standardized and can be validated or qualified in different facilities. With this, having this standardized process also means that there's a better flow for operator qualification, and also that there is a streamlined process for product changeover and ensuring and sharing cleaning validation protocols across different sites. Lastly, we also need to make sure that we can standardize or streamline quality control, going from all of the assays that are needed for assessing the viability of this product, the identity and the cell dose, so we need to have panels that are with the manufacturers of the vectors or the gene transfer models can facilitate having flow cytometry panels that enhance standardized transgene expression assessment across different centers that are using those platforms. We need to standardize our assays for assessing the qPCR assays for assessing vector copy numbers, as well as the assays for assessing the potency of this product. So the cytokine, whether it be cytokine release or chromium release killing assays, we need to have it will be beneficial to the field to have potency assays that are developed, validated, and can be used across different centers with the gene transfer products. The for as we keep developing ways of reducing the time to reducing the manufacturing or the production time for this product, we also need to think about how to regulate the assessment of sterility from both the formal, the USP 71 14-day sterility paradigm, to having ways that we assess interim sterility with the current FDA guidance, including ensuring that for products that are intended for fresh infusion or with a short turnaround time, that there should be interim sterility obtained between 48 to 72 hours pre-harvest. We will still need to think about how to even shorten this interim sterility assessment for products such as the Ultra CAR T or the T-Charge TM platform, where the CAR T cell manufacturing happens in within less than 2 days or within a 24-hour period of expansion. For all of these things, everything comes down to we need to have validated assays that are transferable across different centers. Now, an example of when I talk about standardizing the quality control is a protocol that is available on the Miltenyi with Miltenyi Biotech on the Clinimax Prodigy platform, where you have protocols for protocols with well-designed, well-assessed panels for immunophenotyping of the CAR T cell transduction efficiency and viability using flow cytometry panels. This is available on the website if you want to take a look at what those panels look like. But I give an example here showing the gating with the flow cytometry panels, including the antibodies as well as the gating strategies that are needed to get a clear sense of identity of the engineered T cells. This is what I mean as examples of the gene transfer platforms that are distributed commercially, making available such similar assays for detection of the cell subset of interest. Now, to summarize, the idea behind what I was trying to be provocative about was to imagine an environment where CAR T cell manufacturing is modeled similar to our current transplant programs, where you have the product, which is the transplant or the graft, the transplant product, processed, maybe not necessarily manufactured with the same amount, as extensively as CAR T cells are manufactured, but the processing typically happens at the clinical sites where those products are being administered, including some products that are distributed, as an example, through the National Marrow Donor Program. However, the manufacturing or the processing happens at those clinical sites. And to give you an example, in the earlier slides, I had shown you how many manufacturing sites are available to the current commercial products between Novartis, BMS, Kite Gilead, and Johnson & Johnson. In comparison, there are at my most recent search, there are 227 factor-accredited processing facilities in the US, Canada, Australia, and a couple of other places. This is not counting sites in most of Europe. So there are 227 minimal manipulation processing facilities that are factor accredited for the manufacture or the processing of transplant products. So if you imagine that this product, this same facilities, if they're equipped with with closed, automated, fully automated systems such as the Clinimax Prodigy or the Cocoon, the Lonza Cocoon, this significantly expands the potential for manufacturing of these products at those sites with a little bit of increase in the technical proficiency of the staff at those sites. The protocols are easily trainable to processing, to operators at the sites where where minimal manipulation in particular includes currently even cell selection, such as CD34 stem cell selection on the Clinimax equipment. So, and then even if you think about the centers that are accredited for more than minimal manipulation, which includes open processing in some cases or manufacturing on enclosed systems such as Prodigy, there are still 58 FACT accredited processing facilities across the US, Canada, and some sites in Australia, which I've kind of detailed in the table below. And so I imagine that there is a potential, that there's good potential, because we have this background model to learn from, that there's good potential for developing manufacturing models that are designed around the clinical sites where these patients are being seen and taken care of. The things that we need to think about in expanding this BMT model, this transplant model, to cell therapy or CAR T manufacturing and even engineered TCR manufacturing includes things like the reimbursement models, especially because right now all of these models, or these bedside manufacturing models, are designed around clinical trials, which are supported by INDs. What is the quality oversight? What's the structure for quality oversight for manufacturing across these different centers? In the US and in Canada, we have a model for that, which is the FACT process of ensuring that several standards that are relevant for cell manufacturing or cell processing are met by the FACT accredited facilities. But we can think about these models also in other parts of the world. And then lastly, the regulatory oversight, because again, if we are discussing bedside manufacturing models, then we have to think about at what point are we considering these drugs? I mean, if they are being manufactured at the site of where they are being administered, then we have to think about a paradigm that is different from biologic licensing, because they are not being transported across state lines, as an example. But we still would need some form of regulatory model for those sites that are administering this products, whether it be the registration and following some guidances, for instance, by the FDA, or in Europe by the EMA or other regulatory agencies across the world. But we have a model with the bone marrow transplant or with stem cell transplant that has developed over several decades, and continues to, and we continue to think about these patients who are receiving transplants as being evaluable. We have the CIBMTR where we look at studies of patients receiving transplant products across the world, where these patients are receiving these products that are manufactured or processed at their own clinical site. And so I believe, and I recognize that there are other people in the field, and I hope that there are other people in the field that we can start to have more of a discussion about how to enhance the development and the administration of this CAR T product at the clinical sites where these patients are seen, as a way of increasing access and increasing the rapidity of delivery of these products to patients who desperately need them. I have a slide there with some references that both from the article as well as for this talk. I thank you for your audience, and I look forward to more discussion about this subject. Thank you.
Thank you very much, Dr. Otegbeye, for really lovely and beautiful talk. And here I would like to invite you to join the discussion. Thank you very much. Really, it was beautiful, and I think you have nailed it very nicely, because this is really a big concern in the community, and there are hundreds, if not thousands, of patients who are desperately in need of CAR T cells, for instance. And actually, these waiting lists are quite horrible, and many patients are not making it. So this is why this topic is very important. So we have received a lot of questions, so I'll try to structure them. And first question to you is from Dr. Nayak, who's asking about can you elaborate on the semi-automated models, and which steps are automated and which are not?
Oh yes, thank you for that question. So in the semi-automated models are very similar to really, I mean, so if you think about the open processing of CAR Ts, going from the selection of the starting material, which are the T cells, through to the activation and then the transduction and then the cell culture and then final harvest. So most of those steps can have instruments that can be used to automate the process. So starting with the selection, for instance, I mean, before we had the Clinimax, before the development of Clinimax Prodigy, there was the Clinimax instrument itself that is used for selection or enrichment of T cells or NK cells or various lymphocyte populations. So that selection step in a way it's an automated process as compared to doing like the easy step isolation, for instance, or selection of T cells. And then you also includes things like even the processing, like the washing and of cells using the Lovo or the CPACs, I think, instruments that some other people are familiar with, to even the use of electroporators. The current G-Rex vessels that can be considered in a way, you know, they're closed. Somewhat closed systems. I now have the new generations of G-Rex G-Rex vessels where you can even do the removal of the supernatant and do the cell manipulations or cell processing within that closed system, including the final harvest using the GatherX pump. So all of those steps can be, so we can think of them, but the reason why we think of them as being semi-automated and in a way like partially closed is because you have to move those cells, the in-process material from equipment to equipment, right? As compared to when you have the fully automated and fully closed systems, for instance, like the Prodigy or the Cocoon, where the entire process from start to finish happens in the same instrument. You do need to have some connections for some processes for connecting the media bags, for instance, to the instrument itself. But otherwise, the entire process happens in a fully closed and automated system.
Okay, thank you very much. That was very clear. We have here another question, which I think is a difficult one, but maybe you can give us your opinion. So our colleague is asking about the fact that when you consider these autologous CAR T cells and within the acceptable variability range, the efficacy results are similar. And I'm just quoting our colleague here, which I don't believe is true. But assuming this is true, wouldn't be more relevant actually to worry about the state of the immune system of the patient rather than focusing on all of these quality control steps, which are really taking a lot of time? What are your thoughts? And that brings me to the question whether you believe that having a sort of a bench at bedside model would lead to the same quality of product at the end of the day.
Yeah, so I mean, I think that question is a little bit, there are multiple things in that question. Just as you pointed out, I mean, there is still some discussion about whether if the products are truly comparable in terms of efficacy, right? But then at the same time, when you have discussions about maybe potentially Axicell, for instance, being more efficacious than some other products, they also think about toxicity going almost hand in hand with the increased efficacy, right? Or with the increased trend towards an efficacy, because I can't really say that it's, you know, but okay. But that being said, if we think about a landscape where the efficacy or the impact, patient impact is somewhat comparable across products, and then you're trying to move to a model where you're saying that you want to think about the patient as an individual, and then just trying to optimize getting the product to the patient, is where the bedside model becomes more relevant. But then at the same time, you really can't discount the concern about quality, right? Because while we are trying to be patient focused, we also have to be patient conscious, right? And be concerned about how well can we, if we're seeing adverse events, for instance, between different centers, how much of that can we attribute to a potency issue or something about like the toxicity of the product versus toxicity of the process, right? Which is where being able to really oversee quality comes in. I think it's going to be really hard to get away from having, I mean, between the regulatory bodies that govern administration of these products across the world, you can't minimize the concern about quality, which is why I advocate for having reducing the burden of quality. The one way to reduce the burden of quality or quality control at individual centers is by trying to streamline the process as much as possible. You have flow cytometry panels that are standardized across programs or across products. If your genetic material is your product, then you can develop flow cytometry panels that can be used across different centers. I mean, sterility USP 71 methods for assessing sterility is essentially a worldwide concept, right? And it's a model that we use even for transplant products. So those are not things that we can compromise on, and those are not things that need very high level form of quality control. So I think there is a way to get a good balance between what are the demands we're making of individual centers in terms of quality control, without compromising the need for ensuring that the products we are administering at this site have good quality as well as comparable quality too, because you want to be able to do, I mean, when we look at CIBMTR studies of transplants, part of the reason why we're able to rely on what we get from that is because we know that there is some standard way of assessing what is engraftment, and what is what is graft versus host disease, so as long as we're able to reliably interpret the results we're getting across from different centers, then that'll be the crux of the matter here.
Thank you very much. Again, this is really fascinating, and I believe it's going to be something that we have to consider rapidly. One last question, because we have plenty of questions about small details, how to do this and how to do this. So I'm sure people can reach out to you for these details, but my last question is, what would be your advice, given your experience, to those colleagues, centers, hospitals who would like to embark on such a project of, let's say, bench at bedside model, sort of like homemade, let's say, CAR T-cell product? What to do and what not to do?
I think the first thing to do is to look at your environment or your region and see what the regulatory, who's the regulatory agency that governs your region, and what are the requirements, or what are the guidelines that they have for the type of product you're interested in. So in the US, the FDA has put out a guidance statement for CAR T development. I think within the EMA too, there's been a move to provide those guidelines. So that's kind of the first place to start. The second place is to also reach out to colleagues across the field and see what platforms you can translate to your institution. And how to go about developing that. Are you developing it under an IND? Is it a multi-site? Do you want to look at collaborating with other institutions to develop a multi-center protocol, a multi-center IND that you can adapt to your institution? And then also of course, to think about what infrastructure do you need at your site, right? I think there was a question earlier about what kinds of sites or what kind of centers do you need for GMP manufacturing of these different products. And again, it comes back again to the question of, are you developing a model where you're focused on semi-automated versus fully closed systems? I mean, if it's a standalone facility where you're starting off, thinking about what infrastructure you need for a fully closed system would help you go a long way, right? Because then your requirements for the clean room requirements are different, right? For a fully closed system, the minimum that you would need would be an ISO 8, which I think in the EU is equivalent to a grade D, or grade D or grade C. But then so you want at least an ISO 8 facility to support your fully automated system, like the Clinimax, like the Prodigy or the Cocoon, but then have access to a biosafety cabinet, an ISO 5, which is a grade B or lower facility unit, where you can do all of the connections that you need from the apheresis material to the platform that you're going to be manufacturing on. So I think going from what's the governing regulatory and quality agency in your environment, to collaborating with individuals both within your environment as well as across the world. I mean, right now TCT is it's an international body where you can communicate with different providers across the world, and then thinking about what your local infrastructure is and how you would design it to support your manufacturing in a sustainable way.
Thank you very much. This was really a fantastic start for this year. So I hope everybody has enjoyed this lovely webinar. Again, I would like to express to all of you on behalf of the ICH community, Happy New Year. I'd like to express our deepest thoughts again and sympathy to all patients and families struggling with blood diseases. And may your new year be filled with good health, happiness, and success. So wherever you are, please stay safe and keep well. And I'll see you in a couple of days for the myeloma summary post-ASH 2022. That's going to be on Thursday, the 5th of January, 7 p.m. Paris time. See you soon. Take care.
Thank you.